coming on this Friday to talk about, also to listen to my talk. And I have been asked to give a little bit of an update on the last gift cohort, which I will be doing. I will first do a short introduction about HIV cure in general and what are the main barriers that are still on our road towards an HIV cure. And so first of all, why do we even need a cure, right? We have excellent antiretroviral therapy. We have people right now can live healthy lives almost as long as the general population. You can see this CDC graph that it shows that basically a person without the HIV can live a median of 79 years. And in the pre-ART era, or today, if people that cannot afford therapy, if they are diagnosed at the age of 20, they basically have about 10 to 12 years life expectancy. While a person, so if people take all their antiretroviral therapies, then they are getting closer and closer to the life expectancy of the general population. So we have really done amazing advances in the last decades about treatment for HIV. Also, pills are becoming less and less. So we have like single pills right now. Side effects are less and less, and they are less and less costly. So basically, people can think like people can stay on therapy, and we don't really need to spend much more time, efforts, and money to find a cure. Also, on the another very good news that we came out in the last years is that people that are undetectable cannot transmit HIV to their sexual partner. So basically, this is a very good news and has been so the HIV community and everybody is of course extremely excited about it, and this has brought to the U equal U, undetectable equals untransmittable. And then another good news is that the number of HIV infection in the last years has been decreasing. Overall, 18% less compared to 2010. So basically, there is a lot of nice stuff going on worldwide about HIV. But we still need a cure, and there are many reasons why. First of all, even if the incidence is decreasing, there are still a huge amount of people that are HIV infected worldwide. There are still around 2 million new infections and are over 1 million AIDS-related deaths. And also, the decrease if in HIV infection is not uniform worldwide, but there are still regions where HIV infection is actually increasing. So even if we are doing good globally, it's not everywhere the same. And there are definitively key populations that are more at risk and that we have to pay particularly attention. Also, even if we have drugs that really work well in people that can wear it, the treatment cascade is not yet to where we would like to be. Basically, in our dream, we would like to have a 90-90-90 cascade. And right now, overall, in the US, these are US data, so 75% of people are aware of their status, 79% are on treatment, and 81% are suppressed. This means that basically around 50% of people that are that have HIV are suppressed, and these are not data from the US, but these are global data. But still, like, we are not yet there with our 90-90-90 target, that this is what we will wish to get the epidemics under control. And then another very important point is that HIV still has a lot of stigma, and it's still a mark of disgrace are associated with people, their quality of life, and a lot of people with HIV are discriminated, are marginalized, and despite all of the progresses, it's still perceived as a sickness. So people don't want to carry the stigma around their entire life. And this is why an HIV cure is still necessary, and we are still working hard to achieve it. So there are many barriers to a cure. And we know that HIV reservoir persists during antiretroviral therapy, and it always comes back every time we stop antiretroviral therapy. And so far, there have been one person that has been cured for sure from HIV, even if many people, also if there is at least 2 other people right now in London and Düsseldorf that have had undetectable viral load for a while, and I think there are many more, but we all are familiar with Timothy Ray Brown, or the Berlin patient who has actually lived 11 years post-transplant and it was HIV-free off therapy. And I want to take a minute here to basically acknowledge Tim, who has passed on a recurrence of his leukemia on Tuesday this week. So it has been reported all over the news. And basically, sadly, like 6 months ago, his leukemia came back and he did everything. So he really fought until the end. He did a lot of treatment and was hospitalized for several months. And then I think about a month ago, everything was too much for him to bear, and he decided to go home in Palm Springs with his partner, whose name is also Tim. And there they lived the last months of his life with all of his friends. And he was funny as always. And, you know, like I heard that sometimes he was too sick to talk or to interact, and then he would wake up and he liked to bitch about Trump. Apparently that was one of his favorite topics until the very end. And so, and for those of you who are not familiar with the Berlin patient story, he was diagnosed with HIV in 1995, and he started antiretroviral therapy, but then he was diagnosed with acute myeloid leukemia, which is eventually what killed him as this week because of a recurrence after 11 years. And then he received a double allogenic hematopoietic stem cell transplant. And the doctor at that time, he was very smart, and he decided to transplant the patient with cells from a donor who was homozygous for CCR5 delta 32. And so after a while, when the Berlin patient, also when Timothy stopped antiretroviral therapy, the virus never came back. And he made out of his life, he was an activist. He went everywhere to talk to people, to politicians, to researcher. He has been extremely altruistic with his body, with his blood, and a lot of biopsies were done, and to see if we were able to find HIV. And I was actually looking into it, like HIV has been detected in his tissues, but it just never came back. So basically to the best of our knowledge, he was the one person that was cured with HIV. But of course, what he went through to cure himself is not something that we can do for people that are healthy and otherwise healthy and only having HIV, because of the fact that it's a very dangerous and has a lot of side effects. So we really need to work hard to find a cure that is affordable and available for everybody. And we owe it to Tim and to his legacy to keep fighting for it. So HIV integrates into genome. I think everybody knows now that one of the reasons why HIV cannot be eradicated so easily because it integrates into the genome. And then when people are on antiretroviral therapy, basically it plays hide and seek with the immune system. There is no way that you can differentiate, or the immune system can differentiate a cell who is infected from a cell who is not infected. And because of this, and because HIV usually infects cells who are very long living, the HIV reservoir is very stable. And this is a paper from Silicano from a very long time ago, and he calculated that the stability, or the stability of the HIV reservoir is longer than basically the people's lives. So even if we are on therapy for our entire life, there is no way that we can cure HIV without any interventions. And many groups have also tried to get rid of the reservoir by intensifying the therapy. And this is data from one of our study that Antoine was first author on. And we did not find any difference in HIV reservoir in people who had antiretroviral intensification. This speaks a little bit against low-level replication, which is one of the possible mechanisms that people think can replenish the reservoir. And now this brings me to give you an overview of the mechanism of HIV persistence and basically how does HIV can persist. First of all, if I told you before, it integrates into cells that are long living. So cells can just stick around in our body for a very long time, and then they can also clonally expand. And clonal expansion is one of the way that the reservoir can persist in the body and across tissues. And this is the focus of a lot of research right now. Actually, the NIH is getting out quite a bit of grants and RFAs to really study this particular mechanism, because I think that most of the researchers right now agree that this is the way how the HIV mostly persists. And then, of course, viral transmission from one cell to the other is always possible. There is some skepticism, especially right now, with all of the therapy that we have that is highly effective, and it looks like the virus is suppressed, but there are still people that believe that the virus can replicate in some sanctuary, like for example in the gut or in the genital tract or in the brain, but this is an area of controversy. And the fact that we were not able to find a difference with ART intensification speaks a little bit against this. Another barrier to an HIV cure is that the HIV reservoir is very hard to measure. And in our lab, especially Doug Richmond and his team, people have worked very hard to develop an assay to measure the reservoir. And this is a figure again that everybody probably has seen already from the Silicano lab that kind of showed that the reservoir, also showed 3 ways to measure the reservoir basically. And there is, so you can measure the total HIV DNA, which is the maximum of the entire reservoir, but we know that a lot of this HIV DNA is actually defective. And so basically there is a QVA, which has been the gold standard for a very long time. That is a quantitative viral outgrowth assay, where we can measure virus that is really able to grow in the presence of cells. And this is our yellow circle in there. But then we know that there is also a big proportion of viruses is intact but cannot be induced with QVA. And there is some question in the field, and people realize more and more that this virus probably plays a role in the reservoir. So this makes it even harder for us to characterize the reservoir, because there are not a good and easy way to characterize it. And I know that right now more and more the field is going towards measuring the intact virus, and Silicano Lab again developed this IPDA, which is intact proviral DNA assay. That is a digital PCR-based assay to measure the intact proviral assay. And this is a relatively simple way to measure it. But as you can see, there are limitations to most of the assay. And I think this is something to keep in mind. And this is even more true when we talk about tissue, because of course it's very hard to do a QVA in tissue where the cells are not easy to create cell suspensions. And there is also a lack of biomarkers of HIV persistence. And we could talk about this for a very long time. And I know there is ongoing studies. And for example, Davis MAP study that just ended right now with the ACTG so we are very hopeful that this study might be able to find some clear and nice biomarker of HIV persistence, but so far we don't have anything. So anything useful to characterize or to measure the reservoir in people, also the actual sites of the reservoir in people that are on suppressive antiretroviral therapy. So because of all of these problems and all of these facts, it's very hard to move forward. And this is where we get to the last gift. And the last gift, as one of the main questions that the last gift has been trying to address, is where actually the reservoir hides within the body. And this is another open question in the field, of course, that most of the study that has been done so far are done on blood and not so much on tissues, because of course it's very hard to obtain living tissues from living people. And we can have tissue from animal studies, but a lot of animals, of course, are not people, and so there are limitations on animal studies as well. And there are also autopsy studies, but most of the autopsy studies have poor antemortem characterization, and also the autopsy are rarely performed quick enough to get living cells or to get high quality DNA or RNA or proteins. And to address this limitation, Davy in 2017 actually came up with this idea. What about we enroll altruistic people with HIV who have also a terminal illness, which can be cancer, cardiovascular disease, ALS, or anything else, and have a basically span of life that is estimated to be 6 months and less. We follow them while they are alive very carefully, and then we perform a rapid autopsy at the time of death. This has been done for cancer research and also for some neurological studies, but to the best of my knowledge, this has never been done for HIV research. And what we do, we perform an autopsy within 6 hours from death, which will optimally preserve RNA and protein. And to be honest, to do autopsy within 6 hours from that is not easy. So when we wrote the grant, we probably underestimated a little bit how hard it is to put this together. But we have an amazing team, and I will show you some picture of people that are on call 24/7 and that are able to really perform the autopsy, collect 50 types of different tissues and put together hundreds of tubes and snap freeze them, and they are available in also for research. So why terminally ill volunteers? First of all, because there is no way that we can follow a number of healthy people, a number big enough of healthy people so that we can get the body so rapidly. So basically the advantage of following terminally ill volunteers is that we have the possibility of obtaining their full body for a rapid research autopsy within a short amount of time. Another is that the people at the end of their life, they're very altruistic, and there is no expectation of direct clinical benefit for the study. And in reality, all of these people really manifest the desire to give back to their HIV research field. So the community in general is extremely excited about the last gift because it's one of the few studies that gives the opportunity to terminally sick and old people to participate in each HIV cure research. And so basically this has given us the opportunity to create a new translational research model to advance HIV cure research. Of course, enrolling terminally ill volunteer has also some disadvantage, and this our reviewers are all also very promptly always remind us that people are very sick, that our cohort is pretty heterogeneous, and that sometimes it is hard to generalize our finding to the general population. But despite everything, we believe that the last gift has really helped move the whole field forward, and there are also that the benefit of the last gift cohort greatly outweighed all of also these negative aspects. So the last gift started in July 2017. We aim to enroll 5 participants every year. And the aim of actually the PO1 is to deep sequence and do phylogenomics analysis on the HIV reservoir in blood and across various anatomic tissues. And then on a subset of people who interrupt antiretroviral therapy, we also want to determine the dynamics of HIV rebounding variants and how these rebounding variants actually seed the tissue after that. So you can also see the last gift logo, who has been designed from Andy Kane, who is the chair of the San Diego HIV Community Advisory Board. So this is to say that the community has been extremely supportive of the last gift. And we have a lot of, also, again, a lot of support and a lot of help from them in outreach and to spread the word of our cohort. So here I put this slide in to give you an idea on how we follow people, how closely we follow people. So these are 2 randomly picked participants, last gift 1 and last gift 3. And basically we have viral loads on both of these until, I think for last gift 1 until the day before he died, and for last gift 3 we had it 2 days before he died. So last gift 1 is one example of a person who interrupted antiretroviral therapy before that. So you can see the viral load who is in blue at the end has a little spikes. Just for you to notice this, the timeline is from 1996 to today. So he did had a couple of episodes of viremia, but then he was suppressed since 2013. He was suppressed and then he stopped antiretroviral therapy. And that at the time of that, he had, I think, a couple of 100 copy in his blood. While last gift 3 was basically suppressed. So we have data back until 2014, and he was suppressed his entire life, so for the last 7 years until the time of death, 5 years until the time of death, and he was suppressed 2 days before he died. So you can see the value of this data as a comparison, for example, for CNTN, which is another very nice autopsy cohort. They only have data around in a median of 6 months before that. So you can realize that in the last 6 months of that, a lot of stuff can happen. And so even if somebody was suppressed 6 months before that, this doesn't mean that they are suppressed at the time of death. And for the purpose of characterizing the reservoir, knowing exactly if people were on antiretroviral therapy at the time of death is super important. Here there are data on other 2 people, last gift 4 and last gift 6. So last gift 4 was another patient that interrupted therapy. Last gift 6 actually was suppressed until the time of death. And here is a picture of our rapid autopsy team. Like this, you can see how crowded it gets in our morgue when we work on a rapid autopsy. Like there is between like 10 and 17 people, depending on how many people are available. And how it works, like we have a dinner that cuts the body basically, and then he puts all of the organs on ice in these orange buckets here. Then we have the cutters, and the cutter is mostly me, Davey, Antoine, Steve, or Tom. Like we tend to not let the technician cut just because we don't want them to hurt themselves. So we take that task on, and then we have the people that take care of the tube on the other side of the table. And this is, so we need somebody to be very organized because there is hundreds of tubes and every tube looks a little bit different depending on how we want to, also what kind of tissue we are storing. Like we have flesh frozen tube and we are storing tissue in various buffers. So every tube have a different buffer in there. And then we also collect cells to sort on fresh tissue and to do cell suspension. And this is something that we are still working on right now. Steve Rawlings took the lead to actually optimize that, because I think that the future of the last gift, and this is what we will be focusing on for the renewal will be to be able to collect viable cells for like RNA-seq or for other kind of fancy experiments. Then we have somebody that flash froze the tissue, and this is a very hard job because basically they have to be on their knees for like 3 hours. And then we have people that overview the whole process. And then most of the time we have a couple of runners that just run around and help where is needed. So as you can see, this is a crowded environment. This is a little bit of a problem right now with the COVID going on. We did spend some time thinking about how to change this procedure depending on the status of the body. Like if we have a COVID positive autopsy, for example, we will have most of the team outside the morgue, and our dinner, he has special COVID equipment to minimize the aerosol spread of the virus, like it's a vacuum sew that he uses for people that have COVID, or also for people that we don't know if they have COVID and maybe they died with a pneumonia. So we do have a very specialized algorithm to proceed with the autopsy in the time of COVID. And we are also pursuing actively with grants the possibility to set up COVID autopsy, because there has been huge interest, and we had so many people reaching out to us to see if we are willing to do COVID autopsy, but we will need more funding because setting up a COVID autopsy will require a lot of extra work. So these are the patients who are enrolled so far in the autopsy. So we have 21 people that has been enrolled. Last gift 10 withdrew. So he is our only person so far that withdrew from the study. And we did 11 autopsy, I think. Actually 10. Well, I don't know if it's up to date, but I'm pretty sure we did 10 autopsies. So as you can see, most of our people are older and they are males, but I am proud to say that we also have 3 female and we have 1 transgender female enrolled. Most people are older, but we also got some people that are not so old. Like for example, we have a 50 years old, a 52 years old. So we were, so overall, we have quite a bit of a representation. The diagnoses are pretty heterogeneous, which is a problem, of course, for generalizability of our results. But we have a lot of people with cancer, which I think it's exciting because it opens up a whole new area of research, because there is people interested in the role of cancer and T-cell response towards cancer cells in driving the reservoir. And so we are collaborating with at least a couple of people that are specifically interested in our last gift patient who are dying with cancer. And then you can see people who stopped or did not stop antiretroviral therapy. So for the purpose of the R3, as of our PO1 grant, we were particularly interested in people who interrupted antiretroviral therapy at the time of death. So it turns out most people like to stay on antiretroviral therapy. So far we had 4 people who interrupted and 2 of them died. But most people like to stay on antiretroviral therapy, at least as long as they are able to take it. And we are, of course, still actively enrolling, and we would like to get more people. And so we're, especially right now with COVID, it's not easy. So I wanted just to humanize a little bit our cohort and to talk about how deep is our experience with our study participant. I put a couple of pictures of 2 people that gave us permission to spread their story. So our last gift 1, his name was Tony Bennett, and he died from ALS. And he was our first participant. He died in 2017. And this is a picture of him in hospice with his partner Blake and a cousin. And he was extremely proud to be our first last gift participant. He loved to be part of the study. because he was our first last gift participant, we spoiled him a little bit. I think I visited him probably every day for like weeks. And we became very close, and we had really an amazing relationship. And I still regularly am in touch with partner Blake, and Blake is a fierce advocate of the last gift study. He really still remembers us, and we were the whole team was invited to his memorial. And then this is last gift 5, Mike Danielson. He was another amazing participant. Actually, he also died with ALS, which is a little bit strange because both of us, 2 out of 10 of our last gift autopsy were ALS. But this doesn't mean at all that ALS and HIV are frequently found together. I think there are probably like 30 or 40 cases in the US of people with HIV and ALS. So that's like really a casualty. But Mike was also a fierce advocate of the last gift. He actually talked about the last gift a lot during his life. He executed his right as a California end of life option March 1st of last year. And we were able to recover the body very fast because, of course, we knew that he was dying on a Friday, and we were ready to receive his body. So this was one of our fastest autopsy. And again, with last gift 5, we became very close. He was an amazing person. And they are right now in process to do a movie of his experience as a person living with HIV and being enrolled in an end-of-life study. And the movie is still in production. It's going much slower than I was expecting, but it will be The Terminal Kindness. And I am really excited about that movie coming out. And this is basically Mike with my daughter and my son at the anti-gun march that he joined us. So I also wanted to touch, because we are talking about the psychosocial part of the last gift, we do have an amazing collaboration with Karine Dubé, and she is an ethicist and a psychosocial behavioral researcher, and she has been working very closely with us and the last gift people in the community to work on the ethical part, and this has become a huge part of the last gift. We already have 3 papers published on this topic, and we have 3 more, I think, in press or under review, talking about how people, so how is the ethics of HIV cure research and how people feel about it, how the next of kin feel about it, how the participant feel about it, and how the actual research team feel about it. They interviewed not only all of the patients, also the study participant and their next of kin, but also the rapid autopsy team and the clinical team to feel how people feel being in touch with those people. And it has been overwhelmingly positive, the feedback that we got from everyone. And so for our renewal, we're actually thinking to incorporate the social behavioral and ethical part much deeper in our last gift renewal so that we can keep doing this amazing work. And then, of course, now after half an hour, I get to the actual data. So basically, this is the paper that we published on the Journal of Clinical Investigation, which is the debut paper of our last gift cohort, and we are super proud of it. It has been a huge work from a lot of people. And I wanted to kind of give you a little bit of an overview, and then I let Antoine, so I asked Antoine to be here, so just in case people have any question about it. But basically, going back to our cohort, the first paper was concentrated on the first 6 people that died. So it was last gift 1, 3, 4, 5, 6, and 8. And as you can see, 4 of them were suppressed until the time of death, and 2 of them, last gift 1 and 4, interrupted antiretroviral therapy. 2 patients died on ALS. 1 had a pancreatic tumor, acute myeloid leukemia, oral cancer, and rectal cancer. So what we did from these 6 participants, We sampled pre and post-mortem tissue and blood. We extracted HIV DNA, quantified it by digital droplet PCR. Then we did SGS, so limited dilution sequencing of full-length EMF. And then we basically bioinformatically, Antoine was able to separate the non-intact provirus from the intact provirus, and then from what was the intact provirus, we characterized the HIV, and we did also phylogeographic analysis, which is Antoine's specialty. So these are some data from 2 study participants showing the viral reservoir. So the HIV DNA levels varied across tissue and blood. It went from undetectable to over 600, as of 659 copy per million cells. The median was 56 copy, and the lowest was always in the brain samples. So you can, in overall, we found the highest reservoirs in the gut and in the lymph tissue. This is not unusual. We did find some exception. For example, last GIF3, who died on pancreatic cancer, you can see had a very high level of HIV DNA in the pancreas. So I think that this is not surprising, because if people have cancer, then a lot of T cells, there is inflammation around the cancer, and a lot of T cells might be around the cancer and they carry HIV DNA. So that was an interesting observation. But overall, the HIV DNA level were pretty heterogeneous across the tissue samples. And it is interesting to see that we were able to measure the reservoir in most brain tissue, even if it was low. I think 5 out of 6 patients had detectable viral DNA in the brain. So we were able to recover 605 intact full-length EMF sequences. Overall, I think it was 670. This means that about 10% was defective. This is much lower than what we find in the actual literature usually. But I think that the main reason is because a lot of the defective virus is in EMF. And so by only amplifying EMF, we probably had a bias. We amplified more intact virus. And so we had a bias towards amplifying intact virus, because we know that much more than 90% of the virus overall is intact. We had a mean of 7 sequences per compartment. Interestingly, we did not recover any defective virus from the blood plasma. Also, that's not really surprising, because of course, the virus that is in the plasma is actually actively replicating in people that are off antiretroviral therapy, so it's most likely to be intact. And we were able to sequence provirus from the CNS in 5 of 6 participants. And here you can see a tree with the sequences of the 6 participants, and they, as expected, nicely separate from each other, which means that probably we don't have any contamination. So this is an example of last GIFT1. So this is a tree, with every color is one different tissue. I think we have various observations. First of all, there are intermixing of various tissue, but we also were able to find compartmentalization in various tissue. There was not obvious pattern between compartmentalized and non-compartmentalized tissue, which maybe is just a consequence of our small sample size. We were able to find in various people evidence of clonality of EMF, which again, because we only sequence EMF, it doesn't mean that the whole virus is clonal, but that's still good evidence. And you can see where we have the asterisk, there is evidence of clonality. And this clonality is not only of in one tissue, but it goes across tissue. So we were able to find the same EMF clone across various tissues. And this is again, last GIF1. So most of the virus ended up to be X-tropic, but there is a minority of R5 tropic virus. This is also not surprising because the virus, so these are all long-term survivors, and most of the virus was able to adapt to various tissues. And so this is why most of our participants had actually a predominant of X4 virus. This is a tree of an example of a patient that was suppressed. This is last gift 3. Last gift 3 was very interesting. Again, there was intermixing of the various tissues. There was some compartmentalization. And here there was a huge evidence of clonality across various tissues, at least for EMF. So this speaks either that there is traffic of cells across tissues carrying the same virus, or of course that at the time of seeding of the tissue, the same virus went across various tissues. It's hard to, at this point, to see what was at the origin of this clonal population. So, and now we're going into the phyloanatomy, and this is a collaboration with Antoine and the University of Leuven in Belgium. And this is really where it gets fancy. And I hope that I will be able to explain all of the analysis that Antoine did. And basically, he used Bayesian's statistics to characterize migration across tissue and blood. And this is last gift 1. As a reminder, last gift 1 interrupted their antiretroviral therapy. And you can see that there are various migration between PBMC and all sorts of tissues, back and forward. So here in this figure, you can see all migration pathway that have positive support. This means a Bayesian factor over 3. And okay, and this is people who stopped therapy. And then here is a person, another person who stopped therapy. This is last gift 4. And you see how much there is a crazy amount of migration between tissue and blood. Most of the migration went from blood to tissue, but there are also migration across tissues and from tissue to blood. And some of the migration event also involve the brain. You can see, like, on the left side, you have the source, on the right side, you have the recipients of the migration. And you can see that on the source parts of the migration, we have frontal cortex. This is exciting because basically it is like indirect evidence that the brain might be contributing to reseeding of the reservoir when people interrupt their therapy. So this is another way. So it's again last gift 4, and here it's a little bit less crowded because here in this figure we only show migration pathway with very strong support. This means Bayesian factor over 20. And you can see that there is a lot of GATT migration going on. But still, there is an occipital cortex into our source compartment, which again is interesting evidence that the brain might be contributing to the rebound of the virus. This is migration events in somebody who is suppressed, like last gift 3. So there is a lot of going on even in people that are suppressed, which might be secondary of cells migrating across tissues. And again, there is a lot going on. Of course, there is a lot going on between gut and lymph nodes, but again, there is some brain going on. We are very interested in brain because we are currently working on a grant where we want to specifically look at the contribution of brain and myeloid cells to the rebound of the virus. So we have nice preliminary data that kind of suggests that the brain might contribute. So we then, as Antoine also looked at what factors might be associated with rebound. And what he found was that among many predictors that we looked at, the number of EMF provirus recovered in each compartment, the level of the HIV DNA, the level of viral diversity, and the proportion of X4 tropic provirus were all predictors of migration across tissues. So basically the conclusions of this first study is that there was an emergence of a large clonal intact HIV RNA population after stopping antiretroviral therapy, which seemed to be the source of migration within various tissue across the body. Multiple sites can act as hoops for dissemination of HIV within the host, predominantly blood, gut, and lymph node, which is not surprising, but also brain and other minor tissues contributed to the dissemination. There is viral exchange within brain areas and across the blood-brain barrier, which is particularly interesting for us because we are interested in the brain in particular. And viral dynamics is associated with lower divergence between sites and high HIV diversity at the recipient site. Future direction. So as right now, Antoine, Davy, and I are focusing on the brain and the gut and also the genital tract. These are the three sites that we are actually isolating, and we would like to zoom in and generate more data. So Stephen Rollins wrote his K, and he's about to resubmit it on the genital tract, and Antoine, Davy, and I are working on 2 grants about the CNS and the gut, where we would like to zoom in again and have more information. So we are looking at clonality. That is something that we are very interested in, both with the integration site analysis and T-cell receptor analysis. As I got a grant through NIDA, my Avenir Award, where one aim is fully only looking at clonality. So we are working on that as well. And then, of course, what I am very interested in always is to look at the antigen specificity of the more abundant TCR clones. We are actually working very close with Simon Malal and John Mellors right now, who have some fancy assay where we are trying to figure it out. And then again, we are more and more interested also in the microenvironments around the reservoir, where we look at the microbiome, viral levels, drug levels, and single-cell RNA-seq. So this is all still in process we are working on, and some of these will go into the renewal of the last gift that is coming up. Actually, we are already in year 3. And of course, we are always open for collaboration if somebody has any nice idea. So I would like to acknowledge a lot of people, of course, our last gift participant, our wonderful community, Davy, Antoine, and Stephen, and Karine, and everybody that is part of our rapid autopsy team and clinic as our last gift team in particular. And then, of course, Antoine's collaborator in Belgium that helped us doing all the fancy statistics. And now if somebody has any question, I actually talked more than I expected, so we only have 5 minutes for questions.
Hey, Sarah, there is a question on the chat from Rob. I guess the direction of migration is based on sequencing data, or how was the direction of migration established?
Yes, it is all based on the sequencing data. I let Antoine, if he wants to give you some more details about this, that's all his fancy bioinformatics pipeline. But yes, Antoine, do you want to explain a little bit more to Rosio?
No, you're right. This is inferred from the phylogenies.
So any other questions? You're welcome. Okay. There is no other questions. Well, if somebody has any other question, you can always email me or ask me anytime. And if somebody has any exciting idea how to move forward the last gift, we are always interested in collaborating, of course. We are working actively to renew it and on this R1 RFA that are coming up. So again, like I know as we're collaborating closely with Nadia, for example, like a lot of you are already familiar with the last gift and are already working with us, but we are open to new collaborations.
Well, Sarah, thank you for this great talk. It was a great update. I mean, I think we saw this paper, but you summarized it really nice. It really became more clear and all in one place.
Okay, thank you. Have a nice weekend, everyone.
You too.
Bye.